Hello everyone and welcome to the new session of the lecture series, and today's topic is respiration in plants. And today we'll be solving and discussing NCERT based multiple choice questions. And this lecture is the continuation of the previous lecture, which was again based on the NCERT based multiple choice questions. So let's start with the first question. So here they're asking lactic acid is formed by the process of, so they are talking about anaerobic respiration here. Alright. So anaerobic respiration means it doesn't utilize oxygen. Okay. Now aerobic respiration is also known as fermentation. Now again, fermentation is of two types. First is lactic acid fermentation. Okay, in lactic acid fermentation, what happens? Pyruvate, which is produced, during glycolysis, is converted into lactic acid. Okay, and second one is alcoholic fermentation. Alright, in alcoholic fermentation, what happens? Pyruvate, which is produced during glycolysis, is converted into ethanol. Okay, final product is ethanol, and carbon dioxide is also produced as a byproduct. Now coming back to the options, first option is fermentation. Okay, so this is the correct option. Second option is HMP pathway. What is HMP pathway? HMP pathway is the hexose monophosphate shunt pathway. Alright, this pathway is an alternative to glycolysis. Okay, this pathway is alternative to glycolysis and it produces ribose 5-phosphate, and NADPH. Okay, what are the products from ribose 5-phosphate, and NADPH are the products of this pathway. So this is also not correct. Okay, third one is glycolysis. Okay, this is also not true, because in glycolysis what happens? Glucose is converted into pyruvate. Okay, and then pyruvate enters the fermentation process. Okay, fourth option is none of these. So what should be the answer? Answer is option A, fermentation. Alright. The next question is, in alcoholic fermentation, NAD plus is produced during. So now they are talking about alcoholic fermentation. So let's study alcoholic fermentation in detail. So what happens? Pyruvate, which is a 3-carbon compound, is first converted into acetaldehyde. Okay, is first converted into acetaldehyde, which is a 2-carbon compound. And here carbon dioxide is released, means there is decarboxylation. Now this acetaldehyde is further converted into ethanol. Alright. So now what happens when acetaldehyde is converted into ethanol? NADH, which is a reduced coenzyme which was produced during glycolysis, is converted into NAD plus. Means there is regeneration of NAD plus. So what they are asking question, in alcoholic fermentation, NAD plus is produced during which step? Reduction of acetaldehyde to ethanol. Reduction of acetaldehyde to ethanol. This is the reduction process. There is reduction happening here of the aldehyde group into alcohol group. Oxidation of glucose, okay, this is not true, okay, because NADH is produced there. Oxidation of pyruvate to acetyl coenzyme A, again they are talking of oxidative decarboxylation, and during this NADH is produced. Alright, and hydrolysis of ATP to ATP, this is also not correct. So what should be the answer? Answer is option A, reduction of acetaldehyde to ethanol. Now coming to the next question. Here they are asking which of the following statement about cellular energy harvesting pathway is incorrect. Okay, you have to circle this. Incorrect you have to identify. First one is pyruvate oxidation can only occur under aerobic conditions, they are saying. Alright. Second is autotrophs can produce their own food, okay, but must obtain energy from it by glycolysis and cellular respiration. Alright. This is true. Fermentation usually occurs during aerobic conditions. Okay, this is not true, because fermentation occurs during anaerobic condition. Last option is all of the above. So we have to identify the incorrect statement here. So what should be the answer? Answer is fermentation usually occurs under aerobic condition. This is incorrect. Alright, because fermentation usually doesn't require oxygen. It is an anaerobic process. Okay, talking about the first one, pyruvate oxidation can only occur under aerobic condition. Alright, this is true. In next question is, in the fermentation of one glucose molecule, there is a net gain of dash molecules of ATP. Okay, so again they are talking about anaerobic respiration. So what happens? Glycolysis is the common step, we know. Okay, it's a common step of before aerobic or anaerobic respiration. Okay, during glycolysis what happens? Glucose is converted into pyruvate, and here what happens? What is the net gain of ATP here? Net gain is 2 ATP. Net gain here is 2 ATP. Alright. Actually 4 ATP are produced, but 2 are utilized during the preparatory phase. Okay. Actually 4 ATP are produced. Okay. But minus 2 ATP. Because 2 ATP are utilized or consumed during the preparatory phase. So this gives us 2 ATP. Now further, pyruvate is converted into either ethanol during alcoholic fermentation, or it can be converted into lactic acid during lactic acid fermentation. Here there is no generation of ATP. And what happens? NADH gets converted into NAD. Alright. So what is the net gain? Here is 2 ATP. So what should be the answer? 2 ATP. Alright. The next question is, decarboxylation is not involved in. Decarboxylation is removal of carbon. Okay. There is a removal of carbon dioxide also, you can say. Alright. So now options are electron transport system, okay, or you can say electron transport chain, glycolysis, Krebs cycle, and the fourth option is both A and B. Now during electron transport system, we now know that there is no decarboxylation. There is no release of carbon dioxide. What happens during ETC? There is only transfer of electrons. Okay, there is transfer of electrons from one molecule to another molecule. During glycolysis also, no carbon dioxide is released. During Krebs cycle, yes, decarboxylation takes place twice. Okay, decarboxylation takes place twice during Krebs cycle. Alright. So what happens in glycolysis? Initially we have citrate. Okay, citrate is converted into isocitrate. Isocitrate is further converted into alpha ketoglutarate. Okay. Now, alpha ketoglutarate is a 5 carbon compound. Isocitrate is a 6 carbon compound. So here one carbon dioxide is released. Further, alpha ketoglutarate is converted into succinyl coenzyme A. Okay, this is again a 5 carbon compound. Oh, this is a 4 carbon compound. Alright, and again here carbon dioxide is released. So there are two decarboxylation step here. Succinyl coenzyme A is further converted into succinate. Okay, which is again a 4 carbon compound, and the cycle goes on. Okay, succinate to fumarate, fumarate to malate, and finally malate is converted into oxaloacetate acetate, okay, which combines with acetyl coenzyme A to produce citrate to generate citrate. What should be the answer? Answer is option D, both A and B. Now next question is conversion of phosphoenol pyruvic acid to pyruvic acid, okay, and ADP to ATP is an example of, okay, so they are talking about glycolysis here. So they are talking about last step of glycolysis, the 10th step, during which what happens? Phosphoenol pyruvate is converted into pyruvate. And during this what happens? ATP is produced from ADP. Okay. So now they are saying conversion of phosphoenol pyruvate to pyruvic acid and ADP to ATP is example of photophosphorylation, okay, oxidative phosphorylation, photoelectric phosphorylation, and substrate level phosphorylation. Okay, so what should be the answer? Answer is substrate level phosphorylation. So here what is happening, or what is substrate level phosphorylation? It is the formation of ATP or GDP. Okay, direct formation of ATP and GDP, okay, by transfer of the phosphate group. By the transfer of the phosphate group from a high energy compound to ADP or GDP. Alright. So here what is happening? Phosphate from phosphoenol pyruvate, okay, goes to ADP or combine with ADP to form ATP. Alright. Here you can write inorganic phosphate. Now let's talk about the next question. Which of the following statement regarding pyruvate during aerobic respiration is incorrect? Okay, the option are: it is formed by the glycolytic catabolism of carbohydrate in the mitochondrial matrix. Okay, we know that glycolysis, okay, which involves breakdown of glucose into two molecules of pyruvic acid, takes place in the cytosol or the cytoplasm. But here they are saying in the mitochondrial matrix. So this is incorrect and we have to find the incorrect statement. So our answer should be this one. Second option is: it enters the mitochondrial matrix and undergoes oxidative decarboxylation. This is also true. So the pyruvate which is formed, okay, first it enters the matrix of the mitochondria. Okay, and then what happens? Pyruvate is converted into acetyl coenzyme A. Okay, so this is true. Now third is: conversion of pyruvic acid to acetyl coenzyme A is catalyzed by pyruvate dehydrogenase. This is also true. Alright, fourth option is: two molecules of NADH is produced from the metabolism of two molecules of pyruvic acid. This is also true, because during oxidative decarboxylation, pyruvate is converted into acetyl coenzyme A, and what is happening? NADH is produced here from NAD. Okay, so there are two molecules of pyruvate, so two molecules will be of NADH will be formed here. Alright, here coenzyme A enters and there is decarboxylation. So what should be the correct answer? Option A. The next question is: which of the following is not an intermediate of Krebs cycle? Alright, so now we know that fumarate, okay, is formed from succinate. Okay, then citric acid is formed by the combination of oxaloacetate and acetyl coenzyme A. Succinic acid is formed, okay, from first alpha ketoglutarate is formed. From alpha ketoglutarate, what is formed? Succinyl coenzyme A. And from succinyl coenzyme A, succinate is formed. Alright, and the fourth option is lactic acid. So we have to identify the intermediate which is not formed. So answer should be lactic acid. Alright, option D. Now coming to the next question: which of the following is important in electron transport system? So they are talking about the electron transport chain. The first one is ubiquinone. So now let's talk about the electron transport chain. So first we have the complex 1, okay, which is known as NADH dehydrogenase. Alright, so what does this complex do? This complex transfers electrons from NADH to ubiquinone. Okay, ubiquinone acts as an electron carrier, and from ubiquinone electrons are transferred to ubiquinol. Here it is acting as an electron carrier. Again, let's talk about the complex 2. Complex 2 which is known as succinate dehydrogenase. So what does this complex do? This complex, transfers the electron from succinate to ubiquinone. And then ubiquinone gets reduced, and the reduction product of ubiquinone, or the reduced form of ubiquinone, is known as ubiquinol. Okay, so now here you can see that ubiquinone is very important for the electron transport system. Okay, second option is acetylcoenzyme, which is not correct. Okay, third is dicarboxylic acid. This is also not correct. Fourth option is pericyanate. This is also not correct. So correct answer is ubiquinone. Now, next question is, which of the following processes make direct use of oxygen? Okay, so options are, first one is glycolysis. Okay, so this is not true because glycolysis takes place without oxygen. Okay, it is an anaerobic process or pathway. Next is fermentation. Fermentation again doesn't require oxygen. Okay, third is electron transport. During electron transport, what happens? Or we can say during oxidative phosphorylation. Okay, and when we talk about oxidative phosphorylation, we talk about terminal oxidation. Okay, so what happens during terminal oxidation? Electrons and protons are accepted by molecular oxygen, and what is formed? Water is formed. Okay, so there is direct use of oxygen here. Okay, fourth option is Krebs cycle or citric acid cycle. Here also there is no direct use of oxygen. So what should be the answer? Answer is option C, electron transport. And next question is reduced form of ubiquinone. Okay, so options are ubiquinine, ubiquinol, ubiquitin, and all of the above. So answer is option B, ubiquinol. The next question is, in mitochondria, protons accumulate in the, okay, so let's talk about electron transport chain again. Okay, so what happens if this is the inner membrane? The complexes of the electron transport chain are present in the inner membrane. If this is the membrane, this is complex 1. Okay, we have complex 3, we have complex 2, we have complex 4, then we have complex 5, which is known as ATP synthase. On this side we have the matrix. Okay, and then we have intermembrane space. Here we have intermembrane space. So electrons from the matrix, okay, initially there were high number of protons here. Okay, so now as the electrons flow, okay, from one complex to the other, okay, as the electrons flow downhill from one complex to the other, and what happens? A proton gradient is created along the inner membrane. So protons are, okay, transported to the intermembrane space, okay, from the matrix. Okay, now the protons accumulate here. There is high concentration of protons here. All right. So they are asking mitochondria, protons accumulate in, so what should be the answer? Intermembrane space. Protons accumulate in intermembrane space. The next question is, which one of the following is complex 5 of the electron transport system of the inner mitochondrial membrane. Now we have already discussed that the complex 5, okay, known as ATP synthase, okay, all right, is known as the complex 5 of the electron transport system. Okay, so what does it do? It consists of two parts, F0 part and F1 part. F0 is a membrane, transmembrane protein, or you can say it is a transmembrane ion channel. It's a transmembrane ion channel, okay, through which the protons, okay, flow. And F1 is the peripheral membrane. Peripheral, okay, peripheral molecule or peripheral protein. All right. So, what should be the answer? Answer is option B, ATP synthase. Okay. Now, next question is, in Krebs cycle, the conversion of succinyl coenzyme A to succinic acid requires. Okay, now again let's talk about the citric acid or the Krebs cycle. So what happens when succinyl coenzyme A is converted into succinate? Okay, here what happens? GTP is produced from GDP. Okay. So what should be the answer? Answer is option D, GTP and inorganic phosphate. Okay, in Krebs cycle, the conversion of succinyl coenzyme A to succinic acid require, okay, formation of GTP from GDP and inorganic phosphate. Okay, this is the correct option. Now coming to the next question, which is single turn of citric acid cycle yield. Okay, now again let's talk about the citric acid cycle. Okay, so we already know that 3NADH and 1FADH2 and 1 GTP is produced per cycle. This is produced per cycle during the Krebs cycle or the citric acid cycle. Okay. So what should be the answer? Answer is option C. 1FADH2 is produced, okay, when succinate is converted into fumarate. 3NADH are produced and 1 GTP is produced when succinyl coenzyme A is converted into succinate. And where is NADH produced? NADH is produced when isocitrate is converted into alpha ketoglutarate and further alpha ketoglutarate is converted into succinyl coenzyme A. The next question is end products of Krebs cycle from one molecule of glucose is. Okay, so they're talking about the end product of Krebs cycle. Okay, this is a similar type of question. Okay, we have already done that. So what happens? Per cycle 1 GTP is produced. Okay, and 1 GTP corresponds to 1 ATP. All right. And what happens? 3 NADH are produced. Okay. And what else is formed? 1 FADH2 is formed. And some other byproducts are carbon dioxide, okay, and water. Okay. So what should be the answer? Okay, this is produced per cycle. Okay, this is produced in a single cycle. Now we have 2 molecules of pyruvate, so Krebs cycle will undergo 2 times. So what will happen? 2 GTP are produced, which correspond to 2 ATP. Okay, 3 NADH means 6 NADH are produced in 2 cycle. Okay, and 2 FADH2 are produced. Okay, along with carbon dioxide and water. So what it should be the correct answer? Correct answer is option B. 2 ATP, 6 NADH, 2 FADH2, CO2 and water. The next question is initial step of the TCA cycle. Okay, what is TCA cycle? TCA cycle is also known as the Krebs cycle or the citric acid cycle. Okay, so initial step of TCA cycle to yield citric acid starts with the condensation of, okay, now we know that acetyl coenzyme A, which was produced, okay, by oxidative decarboxylation of pyruvate. Okay, this acetyl coenzyme A condenses with oxaloacetate or oxaloacetic acid to form citrate, which is a 6-carbon compound. Alright, so let's see the option. Acetyl group with oxaloacetic acid and water and catalyzed by the enzyme citrate synthase. Okay, so this is true. Now coming to option C, acetyl group with pyruvate and water and is catalyzed by the enzyme citrate synthase. Okay, this is not true. Okay, third option, acetyl group with oxaloacetic acid and water. Okay, and is independent of the enzyme. This is not true because citrate synthase is involved. Okay, in the formation of citric acid from oxaloacetic acid and acetyl coenzyme A. Fourth option is none of the above. So what should be the answer? Answer is option A, acetyl group with oxaloacetic acid and water and catalyzed by the enzyme citrate synthase. Okay, now coming to the next question, which is which of the following, okay, is correct sequence in Krebs cycle? Okay, so first option is isocitrate is converted into oxalosuccinic acid and then to alpha ketogluteric acid. So now let's talk about the Krebs cycle now. So what happens? Citrate is first converted into isocitrate, okay, with the help of enzyme aconitase. Isocitrate is further converted into alpha ketoglutarate. Okay, finally, alpha ketoglutarate is formed. Okay, which is a 5-carbon compound. But what happens? Initially, an 6-carbon intermediate is formed, which is known as oxalosuccinic acid, okay, or oxalosuccinate. So this is a 6-carbon compound and this is unstable. This is unstable compound and quickly dissociates to form alpha ketoglutarate. Alright, so option A is correct. Option B is alpha oxalosuccinic acid to isocitrate to alpha ketogluteric acid. This is also not true. Then we have alpha ketogluteric acid to isocitric acid and oxalosuccinic acid, which is also not true. Fourth option is, and the fourth option is also not true. So what is the answer? Answer is option A. From isocitric acid, a 6-carbon unstable intermediate is formed. Okay, isocitric acid is also 6-carbon compound, and then finally a 5-carbon alpha ketogluteric acid is formed. Now coming to the next question, which is the chemiosmotic coupling hypothesis of oxidative phosphorylation proposes that adenosine triphosphate is formed because, okay, and the options are high energy bonds are formed in mitochondrial proteins. Okay, so this is not correct. Second option is ADP is pumped out of the matrix into the intermembrane space. Again, this option is not correct. ADP is not pumped. Hydrogen ions are pumped, okay, out of the matrix into the intermembrane space. All right. Third option is a proton gradient forms across the inner membrane space. So this is true. And fourth option is there is a change in the permeability of the inner mitochondrial membrane towards adenosine triphosphate, which is also not true. So what is happening during electron transport chain? All right. We have the inner membrane, okay, where all the complexes of the electron transport chain are present. Here we have complex 1, then we have complex 3, we have complex 2, complex 1, and then we have complex 5, which is known as ATP synthase. Okay, here we have the matrix of the mitochondria, and this side we have the intermembrane space. Okay, so protons which were initially present in the matrix, okay, are transported to the intermembrane space, okay, and due to this a proton gradient forms across the inner membrane. All right, as the electrons flow downhill the electron transport chain, protons are transported from matrix to the intermembrane space, thus generating a proton gradient. So what should be the answer? Answer is option C. Now next question is, what is the function of molecular oxygen in cellular respiration? Okay, so here again they are talking about the terminal oxidation. All right. So what happens? What is terminal oxidation? It is the final step of oxidative phosphorylation. That's why it is known as terminal. All right, final step. What happens here? The molecular oxygen, it accepts electrons as well as protons and it forms water molecule. Okay. So now they are asking the function of the molecular oxygen. So options are, it causes the breakdown of citric acid, which is not true. Second option is combined with glucose to produce carbon dioxide, which is also not true. Third option is it combines with carbon from organic molecules to produce carbon dioxide. Again, this is not true. Okay. Last option is combined with hydrogen from the organic molecules to produce water, and this is true. Okay. So answer should be option D. It combines with hydrogen, okay, from organic molecules to produce water. All right. So this was the end of this session. Thank you very much for listening.